Hello, I'm Kliment from Vilnius University, the Venclovas group, and I will talk about a method that is behind the Voroev group. It's called Voroev GNN. GNN stands for Graph Neural Network. Here is an outline of the method. It accepts protein-protein complex structure, performs tessellation-based analysis, and derives an interface graph from it, and then uses graph neural network to predict per contact quality scores. Those scores then can be accumulated into whole interface score and per residue scores. So firstly, I will talk about how we use Voronoi tessellations to describe interfaces. So Voronoi tessellation is a tessellation of space. For every atom, we assign a region of space that contains all the space points closest to that atom. And some points are equidistant from two atoms, for example. And such points form surfaces called Voronoi faces. We use Voronoi faces as nice representations of inter-atom contacts, and we can compute areas of those faces and use it as weights. So an important point about our approach is that we constrain Voronoi cells inside a solid accessible surface. Otherwise, contacts on the surface of a protein wouldn't make much sense. And we can combine inter-atom contacts into inter-residue contacts just by stacking them together. They form a nice surfaces. And the same can be done with interchain contacts. So here is a small dimer of an insulin, and here is a Voronoi tessellation derived interface representation that is basically a set of contacts constrained inside solvent accessible surface. So this is our geometric representation. Then we derive a graph from it. And our graphs here are quite unusual. And I'm always having problems describing this concept, but basically, conventionally, when you describe protein as a graph, you have atoms as nodes and connections between atoms as edges. Here, we are focused on the contacts. So our graph nodes are contacts, interchain contacts, either on atom or residue levels. And graph edges are borders between adjacent contacts. So here is basically the same picture as before, only colored by different inter-residue contacts. And you can see the borders between inter-atom contacts. And basically, we can take all inter-atom contacts and their borders between them. And then we have this graph of inter-atom contacts, which is quite messy despite the interface being quite small. But we can pull inter-atom contacts into inter-residue contacts and plot it like this. And we can have basically graph of inter-residue contacts. So what do we write into the nodes and edges? So here is a simplified example, our interface representation. And the graph is like this. Nodes are contacts. Edges are borders between contacts, the red lines. And the attributes are contact surface area and contact solvent border, these blue lines for the nodes, and intercontact border lengths, basically those red lines for the edges. And the nice thing about these geometric features is that we can just simply sum them when we go from inter-atom contact graph to inter-residue contact graph. And we also add some information about contact types, basically. And it is done using statistical potential coefficients from our previously developed method, VorMK. So we have VorMK coefficient for a contact between atoms A and B, And also we just consider what would happen if the contact was disappearing and those atoms would contact with solvent. And we also multiply the coefficients by area so that, once again, we can sum them when we go from interatom contact graph to interresidue contact graph. Now, what do we want to predict? We want to predict actually cut score values for interresidue contacts. So we go to the interresidue contact graph, and we can derive ground truth by comparing model, good or bad, to a target structure, and then we can have a cut score value for every contact. Here is the coloring, and basically we want to predict cut scores of contacts. But cut scores of contacts are not convenient things. So we do a pseudo energy trick. So we make them summable, and we weigh them by contact areas, and we make a transformation so that really bad cut scores just penalize the total sum of pseudo energy. So these are conversion formulas. They are really simple, based on the estimate of the just sigmoidal functions, basically. It's a really neat trick that really helps, because we once again have summable things. If we predict cut scores for every intercontact, for every interresidue contact in the interface, we just can sum them to have a global interface score. Now we need data. So we actually produced our own training, validation, and testing set by doing redocking of PDB of heterodimers, non-redundant set derived from PP3D, our server. And the thing here is that we wanted to have a difficult set. So previously our method was good at selecting basically binding sites. So we wanted a set with a lot of almost correct binding sites, but oriented differently. So here is how it goes. Here it is how it looks. So it's basically rigid body transformations, and we have some good models with high cut score and high site-based cut score, but also models with relatively high site-based cut score, but completely incorrect interface. So it makes problem substantially difficult when we do the model selection. And then neural networks. So initial idea for the graph neural network was that we want to train to predict node scores, those pseudo energies. And because we have those nice pseudo energies, we basically train to minimize MSE loss. No need to construct some complex loss function. And we wanted to use both nodes and edge features in some sort of attention mechanism. And then we basically analyze the performance based on how well the global interface pseudo energy sum can select the best models. And so the graph attention version 2 convolutional operator satisfied our requirements. So it basically uses both node and edge features equally. And yeah, maybe it's in a good taste, but I was just in a hurry and didn't paint the picture. It's just in a PyTorch notation. So our ML experiments resulted in selecting a 4-layer GATT version 2 attention network with 8 attention heads. And the final testing before CASP was simple. So basically we compared the performance on a previously unseen testing set in terms of selecting top 1 models and just in general summing the cut scores of selected models. And we compared it with the ideal selection. So basically here it is. So we didn't include perfect models in the set. testing set. And the new method performed much better than the best performing of among our older VORMK-based methods. Yeah, and even if it failed to select top one, the second or the first selected model was on average very close to the basically ideal selection. So, and now a little bit of a case study, if I have time. Here is a target with which, as I was told, basically all QA predictors failed. Here is a model that our method selected. The predicted cut score of the interface derived from our pseudo energy was quite high. So that's why probably it was selected. But if we look at the already published target structure, PDB-ID 7TIL, we see that, well, our method doesn't like it. Why our method doesn't like it? Basically, because in the PDB structure, we have two interfaces. One interface that is similar to the best selected model that we had was actually better in the target. But the other interface is really suspicious. And I'm not really sure what's going on, but I would like to point out that with our method, you can explore such things because we can basically choose what to select, what to look at. And yeah, so this model was over predicted a little bit compared to this, and it shows because the score here is better in the target structure. But anyway, it's a strange case. Maybe it's worth reading a paper a little bit about the structure, and but yeah, no time for it now. So conclusions. If GNN is really very local. So we basically look just at interface context. So it's like both good and bad. Also, VORIF is relatively good at scoring interface, but not really suited for per residue scores that were required by CASP15. And basically, when the method was developed, we didn't really care about it and just did the scoring, per residue scoring at the very last moment. And VORIF is, well, quite unusual. It doesn't use AlphaFold scores. It doesn't use any MSA information, any evolutionary information, but still performs, well, okay. So, and because it's unusual, maybe, and it works, maybe it will be especially useful when combined with other scoring methods. And to conclude, I would like to thank our Venslovas team of 4 people. So Justas Dapkūnas, Česlovas, Venslovas, it's me. I'm thanking myself. So sorry. And Lukas Valančiauskas, our new member, our AlphaFold expert, basically, who produced a lot of models. Some of them were selected. Yeah, and also check out our group's webpage, and also thanks to other members of our lab in Vilnius University Life Sciences Center, and thanks to Research Council of Lithuania for the funding. Thank you.
Let's help Kliment to thank him.